So another very important disease is plague. It is caused by Yersinia pestis, which is a short plump gram-negative bacillus. It was discovered independently and simultaneously by Alexander Yersin and Kitasato Shibasaburo in 1894 in Hong Kong. So it is a zoonotic disease, naturally parasitic in rodents, and is transmitted by fleas, and it is credited for causing at least 3 major pandemics over the past 1500 years. Was also known as Black Death in the Middle Ages. So epidemiology includes the first pandemic that occurred in the Justinian era. Justinian was the emperor in Roman emperor in 527 to 565 BC, and the first pandemic it occurred in 541 to 544 AD, and it began in Egypt, spread through Middle East and Mediterranean Europe. Eventually, whole world was infected, and there was an estimated 100 million deaths. And second pandemic, it was called the Black Death, that occurred between 1347 to 1351 AD. It originated in the area of Black Sea and affected entire area of Europe. 17 to 28 million deaths occurred, and it is believed that 1/3 population of Europe was wiped away by this pandemic. The third pandemic was the modern pandemic that occurred in between 1894 and 1850. It started in Yunnan province of China, affected Hong Kong, Bombay, Madagascar, Egypt, Portugal, Japan, Paraguay, and Eastern Africa, Manila, Glasgow, Sydney, and San Francisco. 12 million deaths occurred in this pandemic. So it is graphical data that shows the reported plague cases by countries, and it shows Africa accounting for the highest number of cases. And the natural cycles of plague includes two cycles: urban or domestic plague, that is, or urban or domestic plague, that is associated with human beings and rodents living around them, and another is wild or syllabic plague that occurs in nature and wild rodents independent of human beings. So vectors are fleas, and they are natural vectors. Reservoirs are rodents, cats, pigs, cattle, sheep, goats, and dogs. Pathogenesis occurs in three forms. Bubonic plague. So bite of an infected flea will lead to an incubation period of 2.5 days, and which develops into a regional lymph node infection. And the lymph node that is most commonly associated is inguinal lymph node, and because of involvement of lymph nodes, it is called bubonic plague. And this picture shows an infected inguinal lymph node. And another is pneumonic plague, and may sometimes be seen in bubonic plague epidemic. Primary forms of pneumonic plague are very rare, spread by droplet infection. Bacilli spreads through the lungs. Hemorrhagic pneumonia occurs, and cyanosis is very prominent. Cyanosis means bluing of hands and feet because of deprivation of oxygen, and bloody mucoid sputum containing numerous bacilli is common, highly infectious, and invariably fatal in untreated cases. So this is a picture showing lung tissue from fatal case of primary pneumonic plague, and you can note the filling of alveolar spaces with inflammatory cells and debris in the picture. So septicemic plague occurs very rarely, and primary infections are rare. It is usually terminal event in bubonic or pulmonary plague. Incubation period is 2 to 7 days. Massive involvement of blood vessels occurs, leading to hemorrhage and gangrene. And this picture shows the blackening of the palm, including fingers. And this is also one of the reasons that it was called the Black Death. A laboratory diagnosis requires containment laboratories with biosafety level 3 and 4. And the specimens are, of course, buboes, sputum, and blood. Microscopy, under microscopy, bipolar staining with Wright or Wesson stain. And the culture requires the incubation temperature of 27 degrees Celsius. Culture is done on blood agar, chocolate agar. Dark brown colonies are formed because of absorption and digestion of heme proteins. The morphology is fried egg morphology. On McConkey agar, non-lactose fermenting colonies are formed. So the serological tests include antibodies to F-I antigen that may be detected by passive hemagglutination, IgG and IgM ELISA, rapid diagnostic assay using F1 antigen, fluorescent antibody technique. And molecular method includes PCR. Isolation from dead rats can be done, and it is done during an outbreak. And 3% Lysol can be used to destroy the fleas that can transmit the infection to the laboratorium or the person collecting for the sample from dead rats.